So welcome to week 8 of our advanced course. So today we'll talk about communication and mentoring. And I think we'll start out with having Charlie talk about Hives for Heroes and all the really great things that they're doing. And if you are looking for volunteer hours or public service credits, I think Hives for Heroes is a really fantastic organization. And so I think we'll talk more about what they're doing and how you can potentially get involved. So Charlie, I'll let you take it away.
Thank you, sir. Again, want to express my appreciation, both Garrett and Heather, for setting me or inviting us, High Star Heroes participation in this event. We're happy. We want to get in front of as many beekeepers or as many folks in the community as we can possibly get. I will blatantly say right off the top, one, I don't have a filter. Number two, I'm going to tell you what we've got, where we've got it, and then what we really need. We're a fine bunch of folks. You guys are a fine bunch of folks, and we want to use everybody's resources together to help a significant part of the community that really desires to be beekeepers. And we need your assistance to do that. Now, what is Hives for Heroes? One, we're a nonprofit organization. We're a 501c3 for those of you that want to know and may or may not care, but we are a nonprofit. Right now, nationwide, we're in all 50 states. We're punching right now approximately 8,000, a little over 8,050 members, all 50 states. Here in Texas, we're at 1,261 folks, beekeepers. We've got a few folks in there beyond that are volunteers. They work with us for various things, or we do business with them. They do business with us. So we've got a lot of that role, but I'm going to focus in just on the beekeeping side of Hives for Heroes. And I'll tell you right now, as an example, here in Texas with our 1,200 folks, 1,200 plus folks, mentoring is a big deal. We really want to, and we're constructed around, and we want to ask everybody to work with us or can we get to a one-to-one-to-one relationship? That's one mentor, one newbie working in one bee yard and learning what's going on. We're talking about pairing experienced beekeepers with experienced veterans, experienced EMTs, first responders, emergency room folks, because they would like to have some relief from the trauma, some escape from being in some pretty crappy situations and need to have the therapeutic value or therapeutic nature of beekeeping explained to them and shown them and worked with them. It takes that eye to eye. It takes that eye to eye capability and that human touch to make that work. Mentoring is a life-changing thing for a mentor. Mentoring, being mentored is a life-changing thing, life-changing experience for that newbie, as we call them. And it's something that we really take a lot of help to get. We need your help to get there. I'm going to pull up a quick map of how Hives for Heroes are, and I'm going to tell you right off the top, it's a big red blob, and I'll tell you why it's that way. And I can give you a little bit more granularity by zooming in and zooming out. Now let's see what we got here. Go there. And then I'm going to go. And Garrett, can you see that?
Yep, I can see it.
Okay, that's Texas. That's every one of those pins and circles and colors. Those are Hives for Heroes members. You can see that big blob that basically runs the I-35 corridor from pretty much down all the way from south of San Antonio over to Houston, up to Dallas, Fort Worth, and all the way up to Oklahoma City. Oklahoma City is at the very top of that. This is I'll say the south edge of San Antonio down here. Red means that those are newbies that do not have a mentor at this time. And whether you want to take a look, here I've just zoomed in on the San Antonio area. Probably about 65, well, actually, it's closer to 80% of the mentor or the newbies in the San Antonio area are looking for mentors. Go up a little bit to Austin, it's a lot less. Go up to the Dallas Fort Worth area, it's more, even more red getting up there. But that's the scope, and that's what I'm doing an ask. I'm really doing an ask with this community, with this group, with your peers, and asking if you have the time in working in your bee yard, which you're already providing probably some mentorship with a member of a local club or elsewhere or somebody else. We would like to ask if we can get your support in being a mentor for Hives for Heroes. Let me get off of this. I'll stop sharing. Come back to this. The pretty pictures. What we do is, I think, is unique. To me, I'm a 30-year veteran of the Army. I've seen a whole lot of crap, been a whole lot of crappy places, and put up with a whole lot of crappy people. This has been probably the most rewarding job I've had. So I have been working with Hives for Heroes, working in Hives for Heroes, for, within, whatever you want to call it. Been working for the last 4 years, and my primary function in there is twofold. One, I'm the Texas state leader. So I'm trying to get all that stuff together and work to get the resources and take care of this and that. But my primary job, I'll be very blunt about it, is finding newbies for mentors and pair them up. I want to have as many possible newbies with mentors to shake their hands and hold them in a bee yard together. And that's what it's seriously about. Now, can we pull it off? Well, I think we can. In some cases, we have a whole community out there of beekeepers that aren't part of organized beekeepers associations, clubs, or activities. Need your help. If you're part of that community that doesn't want to belong to an organized activity, well, we're not really organized. We aren't a club. I'll just flat out say, don't, we're not a club. We work on that one-to-one mentor newbie relationship. We don't have a higher headquarters. We don't have strings and all this and that ties out there and everything, but that's what we do. What we need to have is some outreach to beekeeper out in Uvalde, beekeeper out in San Angelo. We can work with anybody. We really want to have that. Now, I know Garrett said that we'll get to the questions a little bit later, but give you an example. Do you get to pick your own? If you bring that person into Hives for Heroes, yes, you can. But if you, I will pair you with another beekeeper within a 30-mile range or about a 30-minute drive time. Because I want those beekeepers, though, I want those aspiring beekeepers, and even yourself, if you go to their place occasionally to help them out if they have bees, I want them to spend more time in the bee yard than they do in the car. That's just a fact of life. I don't want them to fall asleep coming or going. I want them to be tuned and ready to go. And you check them out in their bee suits and get them in the yard. One of the only rules that we have is mentor's house, mentor's rules. We don't go with that. Now, Hives for Heroes is not a POI. We're not an educational shop. We haven't written our own bee school. We use other people's activities, other organizations. You got one you're sitting in right now. And if you're not a member of Hives for Heroes and you may be wondering, well, how do I pay the bill on this for this course? Let me help you out. If you're a member of Hives for Heroes, what we do is we will reimburse you for the successful completion of each level of a master beekeeping program. So you want to get in there at the bottom floor right now, or you're advanced. Let's roll. Let's roll and get your bud. Go into our webpage, hivesforheroes.org. Sign up. It comes at the top of the page. It says join us, sign up as a mentor, or if you don't think you meet the criteria as a mentor, sign up as a newbie. You sign up, you participate and it doesn't matter whether it's Texas, it's Arkansas, it's Florida, you name it. We've got 14, 15 different master beekeeping programs on our whiteboard that says we will reimburse you for that only if you successfully complete it. So you put your money up front, and there's a thing I like to say, that's skin in the game. That's your skin in the game. You don't want to lose 25, 50, 75 bucks or whatever it is for the coursework plus the exam. We want to make sure you get it. You have a community that will support you. So when you do it, you complete it, send us the receipt, send us a certificate, and we'll either Zelle, Venmo, or send a check. We want you to do well. We want you to apply that with a newbie. So I don't think we can get any better than that as far as helping somebody out. We've got a number of other scholarship opportunities. We've got some other activities that we try to work together and put that out there. We enjoy the heck out of Hives for Heroes. Now, is there any more blatant way to say, I want you to be a mentor in Hives for Heroes? We want you. The I'll say 8 people that are the front office of Hives for Heroes would like to have you help us take care of our nation's veterans and first responders. And some of them may not be bent. They're probably not broken, but they would like to be a beekeeper. Well, our community could definitely use a bunch of fresh faces. We want to continue. We want to grow. We want to benefit the country and its pollination issues. We want to make sure we don't have a food insecurity issue. We want to make sure we've got all that good stuff to make things better for everybody. I'm not saying everything is apple pie, mother. Everybody's got whipped cream and strawberries. I'd like to have that, but we'll try hard. But we would really like to help, and need your help to pull that off. Mentoring is a great god darn thing. I mean, to tell you, having having the ability to show, demonstrate, teach, and somebody learning from you is a way to change everything. It changes their life significantly, and it adds to your life. And that's what we like to see. You ever talked earlier about, again, about um, community hours, community aid kind of things. Hives for Heroes, we're lucky enough to have Hives for Heroes accepted. Your mentoring hours can be captured and then presented as part of your beekeeping journey as a mentor, as a master beekeeper. That's again another. We all work together to come in with the same to work the same mission, and we would really like your assistance to make that happen. Garrett, I think that's enough for me on my soapbox. If you've got anybody that's got any questions, let it rip.
Okay, perfect. Well, if you want to mute yourself and ask a question, but in the meantime, I'll go through some of these questions we have.
Sure.
So Lee Little said, last November I was up with someone, then the person backed out. Should I reach out to them again to gauge your interest or just let it go?
I would tell them to go to connect@hivesforheroes.org. That's me. I'll go back, undo the pairing that I have in my system, and I'll see whether we can find another mentor or another newbie, depending on which side it is, because we have had. I'm there's no BS here. We have had both mentors and newbies ghost the other, and for whatever reason, there's a couple folks that I know have had to bail out of beekeeping because their health was failing, or they were not able to pick a box up and toss it anymore. I've got other folks that just, they signed up thinking everything was going to be great, and maybe they went home and found out that the spouse said, "Bees, no." Yeah, and so they had their significant emotional event, and then they stopped coming, or they didn't show up at all. And here's a problem with technology too. We know that. What happens sometimes is I do a pairing email, send it out. and one or the other team does, one of the other folks on the email do not receive it. So what they don't know that they're supposed to call the other person. So what I've said, to heck with it. Before any pairing gets made right now, I call. I'll call. Most of the time I'm going to be calling the newbies. I want to call, and my own script says, "Hi, Charlie McMaster from Hives for Heroes. Are you still interested in learning about beekeeping? If you are, Hives for Heroes has got a mentor in your area that we'd like to pair with you. Let me know. Just say yes, and I'll get a pairing email on its way to you." And whether I talk to them personally or I talk and leave a voicemail, I've still made, to me, that contact. Now I pull out my little pen and make a mark there and say, okay, I called Bobby and left voicemail, and then I'll go back later. If I don't hear anything within 3 days, I guess they're not going to participate, or they don't want to work it. They don't want to learn about beekeeping. No, no fault, no harm, no foul. Thank you very much. I appreciate your interest and understand that something like life got in the way, or whatever. I don't, no grudge, no grudge held. But that's how desire to do that. I don't want to leave something broken. I really want to make sure we've got contact. We want to have a fix, and we want to do. Okay. Next question.
Yeah, so Doug said, do we have to provide any safety equipment to the new for the new beekeepers?
Well, when you say safety equipment, the first thing comes to mind is the bee suit, a bee suit, bee jacket. We, I will explain. I explained to them that that's another piece of skin in the game. I put it back to the mentor because I want the mentor. It's in our mind. It's better to have the mentor describe what bee suit they would like them to wear. They may have a preference for a full suit. They may not, but we want the newbie to purchase that based on the mentor's recommendation, both style, type, and where might they best get the best deal to get it. But if you have a suit, if you have an extra suit on the nail in the house, and I'm sitting on upstate New York, well, actually, I'm in central Vermont right now, um, a balmy 39 degrees. Um, the If you've got one in the mudroom and you're not using it, and it might fit your newbie, let them borrow it, let them use it, and until they're sure that they want to do it. But actually, the safety equipment, I think bee suit, gloves, add on the gloves, make sure that there's a veil, make sure it's clean, well, clean relative, but make sure it doesn't have any puncture holes so that a bee can get in there. But that's what duct tape is for. But it's the idea of just no, let them get
Yeah,
the
so I
skin in the
mean,
game.
yeah, I mean, basically they're buying the bees, the equipment, everything they need to keep bees based on the mentor's recommendations. So I guess the only thing you need to do is really to be a mentor for that person. Like they're purchasing everything set up, and then you're there to support them.
Well, we're good on that, except one thing. What we'd like to do at the end of that first season, we would like, and this is a would like. It's not a mandatory. It's not a you will. It's a we would like if the mentor would gift the newbie a split toward the end of the year. And that becomes the basis for having it in the mentors, have it remain in the mentor's bee yard over winter. The mentor and the newbie work together so that hive survives that winter. Overwintering a hive, it does make any difference whether you're in, let's say up, way up in the upper peninsula of Michigan or down in Florida. Overwintering bees is a big deal. Overwintering your bees is a bigger deal, and making sure that's your box, that's your colony that you can take after next spring. You get it in there, and you can go maybe bring it out to back to your house or wherever you were going to place bees in your area when you're done as a newbie, when you're finished your first year. Now, we don't stop at your year one, go into year two, year three. Where's the confidence? Build that up with the idea that we would like that newbie to become a mentor in that flow. Safety equipment, like I said, and then the bees. Yeah, they're going to get their own bees. Yes, But that first colony can come from. We would like to have that come from a split from the mentor's bee yard.
So probably for the first year, like, do they typically get their own bees, or is I guess the mentees coming out to the mentor just for the first year to get kind of a feel for it, or I suppose there's different
Yeah.
scenarios.
Well, we like to have. We don't want our newbies to be bee havers. We really want them to know what the heck they're doing, and so you know, I don't say strongly, but we do discourage them from having bees that first year because they'll be bee havers or bee killers because they won't know what they're really doing. And yes, if they do have bees, okay, please mentors go help them out if they ask for it. Go out to their yard. But the primary spot of learning, the primary spot of experiencing beekeepers, that first year for sure is in that mentor's bee yard. We want them to focus on that, and that's the most, you know, as a mentor, you know what your stuff looks like. You know what right looks like with your bees, and you want to be able to show that newbie what right is. And then as time goes on, maybe go out but we don't encourage newbies to get bees that first year.
Yeah, and one thing I'll mention is, everybody that's attending this session, whether you're live or you're watching recordings later on, everybody has at least 2 years of beekeeping experience. And so just try to look back to your first year getting bees and just how intimidating that is. Like, how do you know when to feed, or how do you know when to manage, or how do you know how to do inspection? And so just that relationship is really huge. And you guys are all taking the first step, but the best way to learn and keep bees alive and manage bees is to actually get in the field with an experienced beekeeper and learn all these things. And so I really think this is a really great opportunity, whether you're looking for volunteer hours, but you make a huge difference in somebody's life. I mean, for them to keep bees for longer periods of time and to be better beekeepers. I mean, I can't think of a better organization that helps with this.
Let me add that the best way to learn about beekeeping is to be on the hook with somebody else, having them ask you questions because you don't want to be wrong and you don't want to not know the answer. So it really makes you step up in your own beekeeping uh,
Yep.
knowledge. So it's a really good deal. I highly encourage this.
Yeah. Now, if you've got other friends, you got somebody that's outside the program that wants to be a mentor, ask them to apply. If you want to extend your skills, hey, Garrett's got a great goddamn place. He and Heather are making my life a lot easier by helping us get into talk with you and see you and work with you. And we really do appreciate it. But the ask doesn't stop with you here. If you've got folks that you think should be mentors or could be mentors, and I will tell you that mentors don't know everything. So don't get cocky, guys, folks. Just recall when you were, even after having 3 to 5 years worth of being in the beekeeping business, you know that you don't know all the answers. But you're smart enough to know you don't know all the answers, but you got somebody you can go to get the answer or get an answer. There's no such thing as the answer in beekeeping. It's an answer. So work with that and perhaps a little bit of explanation on our part and your part. We will continue to grow. We'll continue to take care of those veterans and those first responders that would really enjoy having a great experience continue that contributes to our own wellness in the community.
Okay, perfect. And then if you want to share your information in the chat, I guess you said connect at Hives for Heroes,
Yep,
the
connect
email
at
address. Okay.
hivesforheroes.org, and I'll be wide open about it. Telephone number, if you've got your pens and pencils out there taking notes or not, telephone number is 703-624-1337. And why is a damn Yankee sitting in Texas with a Virginia telephone number? Well, I got to see the world. So when I was in Korea, some of a bitch wanted to reach out and touch me full time. And the only way you can do that was to buy me a telephone, and I kept the number. And that was almost 25 years ago. Well, actually, it was more than 25 years ago. It's almost 30 years ago. So The number is mine. So,
Well, perfect. Yeah, and then yeah, I'll send a follow-up email to everybody and then everybody else in the program, and then they can just yeah sign up or contact you for more questions. But yeah, if anybody else has any questions, have your last chance.
yep,
Okay, perfect. Well, Charlie, I really appreciate you joining us today, and I'll see you on the 14th for the apprentice level.
yep,
Um, oh, one last question by Doug.
sure.
Okay, it says I don't have my own yard. My hives are spread out as other homes. Can you pair with new bees that have their own little location?
Probably own location.
Own location.
Doug, can you give me a little bit more explanation about that?
Well, sorry, I was trying to type really fast and catch you. My bees are spread out at various locations, and they're at other people's homes. I don't feel comfortable bringing in a stranger. Are there some newbies that have their own bees?
Um, some of our newbies do, but we really, like I was explaining, we really
I mean,
don't really want to do that, but it can be done.
yeah,
But
okay,
and it, well, we do ask how many colonies the newbies have as part of the or anybody. We always ask our members how many colonies do they have, and we don't really
yeah,
uh, break it asked it for them to be broken out over so many different bee
yeah,
yards locations.
yeah, yeah. I just don't think some of the people that I go tend my bees at would like me dragging strangers in. So,
Yeah, so
thank you.
some folks are against that. Yeah, I
Yeah,
understand that. But yeah, we can what we can do is when it comes time to do that, I can work with both sides and say, okay, it means a telephone call and with you and perhaps a couple of different newbies to find out whether they're capable, if they're ready to do that. But I don't mind reaching out and talking with anybody.
What if like a local oh somebody had a question? I'm not sure that's a question or maybe they're muted. Well, so what if like a local club has like a mentoring apiary and so we can bring them out to the field, so they don't have like or is it more like you want them to come out to like their own yard.
Well,
Let me mute this quick.
nothing's prohibited or nothing's not possible. It's a case. What I'd like to say is we could we could make we could figure that out. I'll just put it like that. We can figure it out because yes, some clubs have learning yards. They have their own training yards or whatever, and they may have static. They already may have mentors in there. The only thing I'd like to ask is if those mentors would become mentors with Hives for Heroes. And I'm not asking them to break the relationship with their local association. What we're asking them is they complement because they may. And here's what when I go out to talk to anybody, any of the clubs, I when I'm talking to the clubs, I'm talking about what they can do for us and what we can do for them. If and I'll put it very bluntly, if we've got 20 people around, um, let's say Greenville, I'll pick up Northeast Texas, somebody in Northeast Texas, someplace in Northeast Texas, Greenville, and we Hives for Heroes have got 10 mentors around Greenville, and they're looking for mentors. The club would be a great source for those mentors. They really would be. And the club could probably appreciate, somewhere between $200 and $300 a year in dues coming in, being infused from the infusion of those Hives for Heroes newbies into their club. So we present that as a win-win. And then with our other bennies that we have, like the reimbursement for master beekeeping programs, that again provides a little bit more for the beekeepers, both newbies and mentors, to continue their growth. But we would like to be in the bee yard of the mentor, but that isn't always possible. Or some folks are more comfortable not having him in their yard, or like Doug was talking about, he's got bee yards out in the field. So, and some folks may not be comfortable letting other people in there other than Doug because they trust him, they like him. They don't know Harris, they don't know Sam. So please don't. Okay, got it. So that's a possibility. Again, Semper Gumby, always flexible.
Yeah, I can add in my own personal experience because I think Chris is even on here when I got paired with um,
Oh, Heather, are you still there? Well, in the meantime, if anybody else has any experiences with Hives for Heroes, maybe there's some people in the chat that have been part of Hives for Heroes, or been a mentee, or maybe they're currently a mentor. Maybe might share their experiences, and talk about really the value they've gotten out of it.
can y'all hear me now?
Yep.
Good.
Okay, it dropped me off. I was gonna say Chris Grover is on here, and so she was a mentee, and I was her mentor. And I think around the time we got paired together, I was in transition of moving, and so I had an out yard, and so I just talked to the guy that owned the out yard, which he was a beekeeper. He ended up being a beekeeper too, so he had no problem with her coming out there, or anybody that I was mentoring coming out there. And then we also had the Longview Arboretum. We had some hives that we were managing there, so we had another source where we could work bees together. So, but that was just my experience.
Now, I know in all cases that sometimes mentor newbie relationships don't work. If it doesn't work, it doesn't work. Let me know though, and I'll work to find you another newbie or another mentor, as the case may be. It's we want folks to grow. We want folks to learn it well, and that means the environment happens to be part of that formula. I don't have any problem with you coming back to the well and saying, well, I need this, or well, I can't do this because my mentor is not available or the newbie, isn't available at the same time. Okay, understand. We will help get that together.
Okay, perfect. Well, Charlie, I really appreciate it, and yeah, to share all your information with me, and I'll send to everybody that's kind of in this 10-week course, but also share it to everybody else in the program. So, I think Monica said she's going to sign up, so that's great, and probably get a few more people on this, but I'm sure we'll have quite a few people across the program that may not be aware of it. I mean, there could be a lot of people that don't know anything about Hives for Heroes, and so hopefully they learn more and then want to be part of it.
Yeah. So, just ask it. The folks here, pass the word out. It looks like we're getting a few. Yeah, we like it. For the participants that are already in it, that are here online, you know, put the word out, pass it out to your clubs. Um, it, you know, Dodie would tell you that, I put too many miles on a rental car and I'll go into the various clubs or wherever. Bottom line of it is that's what's got to be done. We want to stand in front of people and ask, not so much ask for anything, but just ask for their participation, ask for their time. And I know time is expensive, but that's what I think we need to help these folks out in making the community better.
And last thing, do you mind if like we share it and then maybe some other beekeepers can share this recording just to their clubs or people that might be interested in it?
Go for it. Go for
Okay,
it.
perfect. Okay, perfect. Well, thank you very much, and we'll definitely send more information as Charlie provides that to me. But yeah, first, I really thank you for joining us today.
Thank you, Garrett. Appreciate the time. You guys are great.
All right, have a good one.
Take care.
Okay, perfect. Well, yeah, like I said, I'll share more information in email afterwards. So next we'll talk about Tropi mites. And so Hannah Blackburn at the Texas Apiary Inspection Service, she did a recording for the apprentice course yesterday. And so I'm gonna play the recording, but she'll be on afterwards to answer any questions. So this could be a really big problem. Currently, AAA lapse mites are found in Southeast Asia, but if they ever get into the United States, it could be devastating, maybe just as devastating as real, maybe even more devastating. And so, she did a recording kind of talking about the biology of AAA labs and maybe some things that they're doing to maybe prevent it or have a plan in place in case it does get into the United States. And so I'm going to share the recording.
So if you need it, there are a number of QR codes throughout the presentation that will take you to a number of different resources. So some are our office and some are others related to the presentation. So feel free to snag those at your convenience. And I guess we'll go ahead and get started. So my presentation will discuss some of Thailand's beekeeping related to the triple elapsed mite. We'll also discuss the triple elapsed mite and some of their biology, distribution, classification, and things like that, as well as some of the current monitoring and management techniques that are known. But something that you'll hear me say throughout this presentation is there's still a lot we don't know. So even though we do have some idea of some monitoring or management techniques that may have been successful for a project, there is still a lot more that we need to learn about this mite. So take all of that with a grain of salt because the more people research this mite, the more we'll know and the better we'll be able to combat it. Then I'm going to do a brief overview of how Varroa and triple elapsed mites are similar or dissimilar, and then throw some extra resources and tell you how to report it if you think you found it. So in Thailand, there are about 5 species native to Thailand. So the triple elapsed mite is from the Southeast Asia area. Of the 11 species in the world, Thailand has those 5 native species and 1 introduced species, which is Apis mellifera. So they do manage a number of colonies of Apis cerana and Apis mellifera. And these are some of the mites. These pictures are some of the bees that are present in Thailand. So as far as Thailand beekeeping goes, there is a beekeeping industry. They have a number of colonies, about 480,000 colonies, as well as 31,000 beekeepers. Most of the bees that they keep are very calm and docile. They don't wear very much protective gear as far as what we would consider wearing protective gear in the U.S. Their climate is relatively tropical with wet and dry seasons, hot and humid temperatures. With seasonal management, they will have single deeps. They do restrict the entrance because they are concerned with some of those pesky hornets that have been spreading around the U.S. They deal with those on a regular basis. So they do restrict the entrances of their colonies to decrease issues with hornets. And then they keep very little brood. And because of that, they do limit swarming as well. As far as mite management goes, they do treat monthly for triple elapsed mite and the varroa mite. So this is an example of an apiary in Thailand. These photos were taken by Kim Skirm, which is the Massachusetts apiary inspector. I believe chief apiary inspector. I will mention it a little later, but in 2024, a group of beekeepers, researchers, and apiary inspectors went out to Thailand. And so this photo was taken on that trip, as well as the next photo. So in this next photo, you can see how minimal the protective gear that they wear while working their hives is. In some images, you will see people wearing a pith helmet, but in general, most of the time you'll see beekeepers just not wearing anything, just about. And that is related to how docile their bees are. So as far as the beekeeping, honeybee industry in Thailand, these are some of the products that they will distribute and sell. And with this slide, it shows some of the fiscal income from those products. So I don't have a slide that compares like US products versus Thailand products, but you can see that this is about a 38 million US dollar industry in Thailand. So for them, their biggest challenges are finding forage for their bees, as well as dealing with the Tropicalaps mites. Another graphic that I was given is that Thailand beekeeping calendar. So you can see that same image at the top where the beekeeper is not wearing very much protective gear. But if you notice, they treat very regularly and they have different practices very regularly. So October, November, December, January, February through April, May, June, and July through September. That's the entire year right there. They do have a few flows. So they'll have the wildflower flow, the lychee and longan flow. And then there are a number of treatments that they use. They do usually incorporate 1 to 2 brew breaks. They'll treat with 6 different mite treatments and they'll adjust those throughout the year. Then they do have 2 big honey flows where they pull honey weekly. And then compare that to the US northeastern US beekeeping calendar. So 1 to 2 brew breaks, that sounds familiar. But with the US beekeeping calendar, they'll have 3 colony buildup periods where the colonies just grow and grow and grow. There are about 3 mite treatments, depending on the beekeeper, that may be applied, and 2 big honey flows. So if you notice that Thailand slide showed something almost every single month going on, whereas with this northeastern US beekeeping calendar, there's bigger spaces in which these things occur. So what about Tropicalaps mites? So Tropicalaps mites, there are 4 known species of Tropicalaps mites. Previously, there were only 3, but I believe it was 1961. Tropicalaps mercedesi was distinguished from Tropicalaps clarii. Tropicalaps mercedesi and clarii are important whenever people talk about Tropicalaps mites. Generally, they're talking about these 2. But for the purpose of this presentation and for the purpose of most of the warnings related to Tropicalaps mites, you're going to be hearing people talk about Tropicalaps mercedesi. Both of these mites did originate in Apis dorsata, so the giant honeybee, but as time has passed, they have found their way to Apis mellifera. This is relevant because Apis mellifera is what we have in the US today. So basically, they have migrated from Apis dorsata to Apis mellifera. Apis dorsata generally have a seasonal migration between November and January, which can help control those Tropicalaps mites that are present. Additionally, they build single combs along a tree line, which on my next slide, I'll have an example of that. Whereas Apis mellifera, they are moved for pollination seasonally, but as far as them building their nest, they have multiple combs within a colony. So in 1961 or thereabouts, it was established that Tropolaelaps mercedisi had moved from Apis dorsata, which means they still occupy Apis dorsata colonies, but they were observed in Apis mellifera colonies. One of the observations was an association with field rats, but those rats were associated with honeybee colonies. And so that it was one research paper and it mentioned it briefly. There is, yet again, more research that needs to be done on this topic, but that is something that led to the realization that they were in Apis mellifera. So as far as the Apis dorsata, you can see on the left where these Apis dorsata colonies will just, each of those frames, each of those layers of wax is its own colony. So these are all sister colonies. And it is very common to see Tropi mites wandering between each of those colonies. So they are quite mobile as far as mites go compared to Tropi mites. Compared to Varroa mites, the Tropi mite is quite a bit more migratory and movement friendly than that of Varroa. So they can be seen going from colony to colony to colony along those tree limbs. That is relevant because of the way we keep beekeeping, the commercial beekeeping today. Even hobbyist beekeepers have their colonies somewhat relatively close to each other. So it would not be uncommon or not visible for the Tropi mites to move from colony to colony to colony within a yard if they were to arrive. Now, as far as mite dispersal goes, there is some record of it happening during drift or robbing or foraging or potentially even mating, but most of the dispersal is believed to have occurred by human means. So moving frames of comb from one colony to another, rotating boxes, moving colonies for pollination purposes, or even selling these to other individuals. This map shows the current known distribution of Tropi mercedisi in its association with Apis mellifera. So they originated out in the Thailand area, Southeast Asia area, but with more recent records, it has been found that they have made their way to Georgia and Russia, which is a good little ways from Thailand. Not U.S. Russia, but not U.S. Georgia, but they have made their way to Georgia and Russia, and that report was distributed in 2024. So this map is starting to grow. The longer time passes, the more red you're noticing on this map. With that, you can see the spread of the Varroa destructor mite. So they started over on the right, and they were in 1952, 1950s. They were in Japan, China, and Russia. But by the 1980s, they had made their way to the US and Canada. So even though the Tropi mites have not been dispersing as quickly as Varroa mites did, we are keeping an eye on this pest because we do not want Tropi in the US. Nobody wants Tropi in their area, to be honest, because of how devastating they can be to colonies. So, with that in mind, a group of beekeepers, bee researchers, and apiary inspectors went to Thailand in 2024, funded by Project APISM. If you are not aware of Project APISM, look them up. They are a great resource. And I will have a link to one of their YouTube channels later in my presentation. But in early 2024, a group of apiary inspectors, beekeepers, and bee researchers went to Thailand to learn about AAA lapse mites. A lot of the slides and information that I will be presenting came from that trip. So their intent was to go learn about AAA lapse mites, get some of these apiary inspectors to know what they look like so that they could come back and inform other apiary inspectors and get us on watch. If we don't know about something, we can't be prepared for it. We can't look for it. So the following November, so November 2024, they hosted a AAA Labs workshop in Auburn, Alabama. And I was one of the apiary inspectors that was allowed to attend. We discussed what AAA Labs was. We watched a lot of videos. We had some hands-on with dead AAA Labs mites, not live ones. No one is bringing live Tropi into the U.S. But we had some hands-on so we could look at the debris samples and do a few other things. And then we also discussed about what are we going to do if AAA Labs makes it to the U.S. So this next slide, there is a AAA Labs mite wandering along the frame. So if you look, it's just, I have a few videos throughout my presentation and I added these so that y'all could see what they look like. You can see how quickly this mite moves across that frame. There's very fast movement. It kind of stops and starts, but it's rather quick. So this next slide has a honeybee with a AAA lapse mite arrowed in red and a varroa mite arrowed in blue. And you can kind of see the size difference, but I will have a better example of the size difference later in my slide set. As far as the life cycle goes of AAA lapis mites, they reproduce much faster than varroa mites. So this graphic is from the National Bee Unit in the UK, and I thought it was a great resource, so I got their permission to share it with y'all. Maggie Gill is a researcher for them, and she does a wonderful job. So this shows you the difference in how many mites are produced, either in a worker cell or a drone cell, over that period. So basically, most cells have one mite, one female mite, but can host several. AAA lapse and varroa can coincide and share a cell together. And you'll see in a later video of some of the reactions to them sharing a cell. As far as AAA lapse mites goes, they will enter the cell in the third, fourth, or fifth instar, so between days 6 and 10. But they will oviposit after the second day in the cell, and they'll lay a new egg every 24 hours. Basically, they will produce about 2 to 3 mites from a worker brood cell. So in this graphic, it shows 3 mites for AAA lapse versus 1.6 mites for varroa in a worker cell, and 4.5 mites in a drone cell versus the 2.6 in a drone cell for varroa. This next slide, it's more for your information than something that you'll have to look for. These images are taken with a microscope. So if you don't have a microscope, it's not the end of the world. But I just wanted to show you some of the life stages of the AAA lapse mites. So those deuternymphs in adults, those last three at the top, will be the ones to emerge from the cell. Once the workers emerge and or the drones emerge. Then at the bottom, you can see the difference in the sexes of Tropilaps mites. So this is getting into the nitty gritty, but I thought it was cool, and I like sharing it with beekeepers whenever I discuss this. You can see how that plate along the center of their body doesn't fully connect for the males versus the females where it connects fully. So on this next slide, we have some debris sample. And you can see that Tropilaps are circled in red. So two of those are really, really light colored. Those are deuternymphs. But the rest of them kind of show up a little bit better. You can see that the ones that are upside down, you can see the plate down the center of their body, as was shown in the last slide. That is one very helpful way of noticing them on a debris sample. I had the opportunity to look at some debris samples, and with all of the wax and this and that and the other, you have to have pretty good eyes to notice them on a bottom board debris sample. And then we also have that varroa mite circled in blue. As far as biology and behavior, so with Tropilaps mites, they are not known to be on the bees for very long. So they are known to spend anywhere from a few hours to a few days versus the period on the bee, the phoretic or dispersal phase of Varroa, where it's 3 to 13 days, which can be an average of 7 days. So if you compare 7 days to approximately 1 day, that's a big difference in the period of time it takes them to go from emerging from a cell to being able to go and start reproducing again. They can be transferred from adult bees or seen running along frames during this period of time. At this point, the best we can tell is that they lack the morphological adaptations, the mouth parts, to feed on adult bees. So that's part of the decreased phoretic phase is with the varroa mite, they actually have to have a period on the bee where they feed before they can go and start laying eggs. Well, that's not the case for Tropilaps. Additionally, one of the benefits or good things that has been researched is how long can they survive without brood? At this point, they need brood to survive. That is what we know. If they are going to go from cell to cell to cell reproducing, they need that brood, those larval and pupal bees for feeding and to lay their eggs and so on and so forth. So one of the things that was looked into was, well, how long can they survive without brood? Because if they can't survive without brood, then we've problem solved. We just go broodless for a while. But one of the things that was found was, as far as surviving on adult bees, they can only survive about 1 to 3 days. Surviving on comb or other hive materials, it's about 5 days, a little bit more than 5 days. Surviving on dead bees or dead pupil would be approximately 7 to 8 days. So this is good news. This is not bad news. This is good news. But then there's that reproductive phase where they have to be under the cell. It's believed that they consume the hemolymph of larvae and pupa, but more research is still needed on that front. They do prefer larvae that are at least 3.5 days old. They use multiple sites to feed. So this is another relevance as to why Tropilaps are not that great, not so good for bees, is they will have multiple feeding sites. So if you look at the image on the right, you can see those black dots on that larva. And those black dots are each of them feeding sites. With triple alaps, they feed multiple sites, but with Varroa, they'll open one site and continue feeding from the same site. So you'd never thought you'd hear me say this, but in some cases, Varroa is the nicer cousin of the mites. So with Varroa, like I said, they feed one site. Tropicalaps, they feed multiple sites. This can lead to deformations in the bee once it emerges as an adult. And then additionally, there is no significant preference that is known between drone or worker brood for Tropicalaps mites. Then oviposition. So they do not need to feed before they can start laying eggs. This is another bad thing about them. And they prefer larva or pre-pupa, but will lay on pupa as well. So here are some lookalikes of the Tropicalaps mite. If you notice on the left-hand image, we have a few different critters here. So we've got our regular old Varroa mite, our Tropicalaps mite in the bottom center, and then a pollen mite and a Berula fly. So the pollen mite looks more like the Tropicalaps mite than any of these others to me. But if you notice, it's more circular versus the oval of Tropicalaps mites. Now, Tropicalaps mites and Varroa look similar to a certain extent because they have that oval shape. Then on the right, we have male and female Varroa mites, and there has been some comparison as far as appearance of those two Tropicalaps mites. So we have our size comparison. What does Tropi look like compared to Varroa? Well, this is to scale. We have a Varroa and a Tropi side by side. And then at the bottom, we have the US penny with those two side by side. Then finally, the bad news. If you have trouble reading the 0 on a US penny and the date, you're going to have trouble seeing Tropicalaps mites. They can fit within that 0 in a US penny. So if you have a US penny next to you, have a look at it and see how small that is. So this next slide shows an image from Project APISM, as well as a video from the National Bee Unit. What's relevant in this video is you can see those front two legs moving as if they're antenna. So that's a really cool feature. They use their front two legs for examination of things. So let's go into the visual identification and morphology and things like that. So morphology, the bodies are longer than they are broad. They are about 1/3 the size of a Varroa mite. So 1.5 millimeters in length and 0.5 millimeters in width. They do have that light reddish brown color, and there are 8 legs, which can be visible from above when they die or when they're moving. But as I mentioned, those front 2 legs, they use similar to antenna. They have their own antenna, but they will use their front 2 legs similar to antenna for sensory detection and things like that. If you're looking for them on a frame, you're going to look for fast moving objects. So if they're on the brood frame, you can see them on the surface of the comb. They can be found throughout a colony. Another thing that I will show more videos of is they have a stop-start motion where they move, stop, move, stop, move, stop. So that is indicative of the movement of the Tropicalaps mites. If there are high infestations, you may find them on adult bees. And when they're on the adult bees, they're not commonly found between the abdomen and the thorax. So then if you're looking for damage, the damage is very similar to that of Varroa. which is unfortunate because it would be nice if there was a distinction between the two types of damage. But to date, the research has found the damage is very similar to that of Varroa. You'll notice the spotty brood pattern, chewed down brood, bald brood where the cells have been uncapped close to the emergence of the bee. Something else you may notice is workers dying at emergence with their proboscis extended, so with their mouth parts extended. Additionally, you may notice deformed wings because they do spread deformed wing virus, as well as other deformities based off those multiple feeding sites. And then there may be mite bodies in the bottom board debris. So here you can very clearly see that stop-start motion. They move, they stop. They move, they stop. And you can also see those front two legs being used as antenna. I do have a few videos within my slide set because I find this very interesting. I find this very, the more you see it, the easier it is to call it out. So I wanted to include some of these videos in my presentation. So this next video is from the UK National Bee Unit. So you can see there's the gravid female, the fat female. She is harassing a few of those Tropolaelaps mites, a few of those Varroa mites. And it's found that Tropolaelaps kind of tend to do that whenever they're sharing a cell with a Varroa mite. And then if you notice the bottom center, that mite is not pregnant, whereas this other mite has that extended body. She is gravid. So as far as Varroa versus Tropi, the movement is slower for Varroa than Tropolaelaps. So Tropolaelaps move quickly with that stop-start motion. Tropolaelaps can be found throughout the hive, so all along the comb, versus with Varroa, they tend to stay near the brood frames. Tropolaelaps have a very short phoretic phase, anywhere from an hour to a few days, compared to that 7-day average for Varroa. Tropolaelaps at this time, with the research we have to date, are only known to feed on the brood. And it is believed that they feed on the hemolymph of the brood, whereas Varroa are known to feed and survive on adults and brood and focus on the fat body. But as I said, more researches need to be done on a lot of different topics. Tropolaelaps can oviposit without immediately feeding, whereas Varroa need to feed before they can oviposit and lay eggs. More research is needed on monitoring, which I will discuss monitoring here shortly. There are genetic markers for miticide resistance with Tropolaelaps mites. So we do know that Varroa can and have developed resistance to some miticides. But of that research, it wasn't done on Tropi. But Tropi, we have found genetic markers for miticide resistance. So that is something that could potentially come down the pipeline. Tropolaelaps are known to vector many viruses and possibly others that we're not aware of. And lots of the viruses are very similar to that transmitted by Varroa. Colonies collapse much faster, and I do have a slide later in my presentation that kind of gives you a timeline. They double their population in about 7 to 15 days versus that of Varroa, which take about 30 days. And they reproduce 2 to 3 times faster than Varroa mites do. So here we have another video. And like I said, I'm going to have a few videos here, but some of these videos show, oh, here's a good one. You can see where they've got that chewed down brood, some melted brood, and those are some of the indications that you could have a Tropolaelaps mite infestation. So that's a good representation of those. Here we have a mite on a queen, a mite next to pupa, And you can see how it blends in so well with that wax. So as far as monitoring goes, these are some of the techniques that have been used to check monitoring efficacy. So with Tropi Labs monitoring, they did visual inspections on adults, visual inspections on brood comb, and visual inspection for bald brood. In Thailand, usually they said if they see bald brood, they know it's probably time to treat. That they probably have Tropi mates in their colonies. Additionally, one of the research, or a few of the research actually, have done uncapping of the brood. Now, this is a very great way of checking for Tropi mites. You look actually in the cell. The problem with that is it does take so long to conduct the uncapping. Like usually you're taking capped brood and you're uncapping about like 100 cells or more. And that takes time. And then even once you get it uncapped, you have to be able to see the mite. With the alcohol wash or powdered sugar shake, because of the decreased phoretic phase of Tropi mites, that's not as accurate a method for monitoring, but it was looked into. Then there's the sticky board, the checking the bottom board debris. Oh, excuse me. And then other things that were checked were the bump test, where you take a frame with capped and uncapped brood, you shake the bees off, and then you take that frame and you bang it. So if you notice the tray on the bottom right, that's an example of the debris left over from a bump test. Basically, you take that frame, you bang it multiple times on both sides to try to dislodge any mites or anything that you could then look at under a microscope and check for mites and things. Finally, they also did eDNA testing, where you would take a swab or a Q-tip of sorts and swab the entrance and some of the cells to see if there's any indication of Tropi mite DNA on the frame. So here is another one where that mite just blends right on in with the frame. And then we have this video here. You can see where they have uncapped and like they've got that bald brood look to them. Some of these that are kind of raised up a little bit, but that was just another video. So as far as some of the Tropi mite damage, we have brood reduction and decline of development, which, I mean, we're not surprised there. That spotty, irregular brood pattern, chewed down brood, bees emerging with their proboscis or tongues extended, dead bees, deformed bees, either wings or otherwise, bald brood or uncapped pupa, which you can see a few of those on this image here. Holes in the cappings. So some of those cells, you'll notice that the bees have like opened it up a little bit and didn't bother to close it back up. So that's what that's talking about. There may be some absconding of colonies with Tropi mite infestations, as well as lowered honey yields. Additionally, since wax moth and small hive beetles are secondary pests, it is known that wax moth infestations increase. I believe that if we had Tropi in the US, especially in Texas, we would notice more small hive beetle infestations. And then this next slide actually shows a comparison of some of the viruses associated with Varroa and Tropi. So this is just one set of samples, but in that set of samples, you can see some of the similarity between the viruses that are present in both mites. So this next slide shows it's a medium infestation to my understanding, and you can just see those mites all over the place. You can see that stop-start motion. You can just see them moving around. So as far as those monitoring techniques, so as mentioned earlier, as uncapping pupa was 92% effective, but it's time consuming and hard to see the mites. The powdered sugar shake and alcohol wash were less than 40% effective when checking for mites because of that short phoretic phase. The sticky board, 100% effective. Great for detecting them, but it's time consuming and hard to see the mites. The bump method, which we use in the USDA National Survey and have used for years, did come out to be 73% effective, which is great. But it does kill some of the larvae. Finally, the eDNA method is very effective, but at this point, there's very few labs that offer it, and it's quite costly at this time. So as far as management methods, so we've got some non-chemical methods listed here. But of those, making splits or brood breaks was the best method for Thailand beekeepers. It's their golden standard. So there was some research in the beehive thermal heating pads, but more research is needed before it can be considered a great method. Additionally, we have some of those chemical methods. So formic acid and thymol were both in the Apiary Inspectors of America research. Both came up as great methods. But I have been asked a number of times about oxalic acid use. And I did find that in the National Bee Unit, they found that oxalic acid vaporization or sublimation was somewhat effective. It was less than 70%, which isn't great because when you think about the fact that if one mite survives, she can start the infestation all over again. Less than 70% still leaves 30% of your mites. But it was something that was mentioned, something that has been researched, and I knew it was something Texas beekeepers were going to ask about. So on this next slide, we have day 1 versus day 20, and you can see the decrease in bees. You can see that spotty brood pattern. And these are videos from the National Bee Unit. They were real gracious and shared some of their research, some of their videos with me for my presentation. So we're getting towards the end of it. Are Tropilelaps mites really a problem? And the conclusion that we've come to is yes, they will be. And they are currently in the sense that they're expanding their geographic range. So all it would take is one Tropilelaps mite to make it to begin an infestation because of how fast they reproduce and how quickly they travel. They do reproduce faster than Varroa. They feed exclusively under the wax capping, but they move very quickly. And with their feeding, they do leave bee deformities, scar tissue on those adult bees as after they've emerged. Another problem is that the damage is very similar to that of Varroa mite. So even once we got Tropilelaps mites in the U.S., which it is assumed that they're coming and is a foregone conclusion. They're not here yet, though. But once they were to arrive, detecting that damage would be quite difficult because of how similar it is to Varroa damage. They do spread viruses and weaken colonies. And they wipe out a colony if they're left untreated. And that's my next slide. But as far as monitoring techniques, they're very hard to detect, and we don't have a lot of great techniques out there. And management, we do have the splits or brood breaks as far as non-chemical treatment. But as far as chemical treatments, there's not a lot of options out there for us. So those are some of the things. In Thailand, when they were asked, how long will a colony survive if left untreated? They said about 1 month. Just 1 month. So here is a slide with some of the additional resources. That QR code will take you to the Tropi stock webpage. And they've got some great handouts. They've got this debris sample, like test yourself postcard. And on the back of that, they have the QR code with the answer key, just in case you need it. Additionally, we have this QR code for Project APISM's videos. During that trip to Thailand that they funded, they actually sent out a videographer and recorded a number of great videos, and I highly recommend checking those out. Some of the researchers that I found, followed and learned from, Jeff Williams and Rogan Tokash were part of that Auburn workshop that I was able to attend. I have heard seminars from Dr. Samuel Ramsey as well as Dr. Jeff Pettis. And then Dan Etheridge and Maggie Gill from the UK's National Bee Unit were very gracious. They've got some videos on YouTube of some of their research. They even recently did another trip to Thailand to do more research. So I see more coming from them in the future. As far as more information is needed, that is absolutely the case. There is so much we do not know about this mite. So let's get to what to do if you think you found it. Report it. Absolutely. Call our office, send us pictures or videos. We Depending on what we see, we can come out and collect sample, either debris sample, wax, a variety of different substrates. And then we can store it and send it off to the USDA. There is the understanding from across US with the USDA is they would prefer that those items are collected by an apiary inspector instead of going outside of the chain of command, basically. So if you suspect it, absolutely report it to our office. We are more like we were receiving the quote unquote murder hornet reports, and they all turned out to be cicada killers, but we would much rather know than not know and allow it to spread. So what is Texas doing to stop AAA lapse mites? Well, we are working with Apiary Inspectors of America, the Texas A&M AgriLife Extension Service, and other groups, and discussing different things. We have had the opportunity of attending that workshop in Auburn in November, as well as the January 2025 Apiary Inspectors of America meeting, and where we discussed planning. What are we going to do if AAA Labs makes it here? We're not trying to sit and wait for it. We're trying to be proactive. Additionally, I am doing public talks to different bee clubs. So if you know of a bee club that is interested, have them shoot me an email. My email will be on the last slide. And I'm happy to line something up to discuss AAA lapse mites to different bee clubs. I've done a few talks already, and I plan on doing quite a few more. Additionally, we are doing the USDA National Honeybee Survey, where we do that bump method to try to detect AAA lapse mites. And I'd be happy to discuss that with anybody. So thank you very much for your time. And if there are any questions, I would be happy to address those.
Okay, perfect. I think Ken is on the call, right? And so if you have any
Yes.
questions, yep, you can answer any questions.
So in the chat, someone said, I wonder if they've tried bringing a mite biter bee breed. To my knowledge, they have not tried that. There is an industry in Thailand of beekeeping, but it's not like the US industry at all. They don't do all the breeding and this, that, and the other that we've had a tendency to see all across the US. But it is something to take into mind, take into consideration. Would that help decrease the mite populations, decrease Tropi potential of getting here? Another question was, will this recording be available by itself or will it be embedded in today's class recording or her slides? Thank you. So What's the plan for the original recording,
Yeah, so
Eric?
the one from yesterday, we had that all separated. And so if you want to share that, I can send you the link. And Hannah can also provide the slides too that I can link. And so yeah, I mean if Hannah's okay with it, we can send out the information and you can share with anybody in your club or any other beekeepers.
And I am doing talks for clubs. Um, so I'd prefer that my slides didn't get shared with like outside of the Master Beekeeper program for now. Just because I'm weird like that. Um, I the way I view it is it's not my research. I'm just presenting other people's research. So I'm paranoid about having my name associated with all of the hard work that they did. So Ski Dodson. Hannah, Canada imports bees from Asia. What restrictions do they have on Canada? So we don't get bees from Canada. Um, and once Canada found out about Russia and Georgia, they cut off imports from Russia and Georgia. Once there was official report of Tropi in Russia and Georgia, they stopped purchasing bees from there. But as far as like the US importing bees, we're not supposed to be importing bees in the US, my understanding.
Well, perfect.
Were there,
Are there oh, you can go.
I was just going to ask if there were any other questions.
I don't see any other ones, but if anybody wants to unmute themselves, feel free to ask any questions. Okay, perfect. Well, I don't see anything, but if you have any questions after this, I think Hannah can just put her email in the chat just so you have it, and she'd
I
be
can do
happy to
that.
answer any questions. Okay, perfect. Well, I think we'll take a 10-minute break and then we'll talk about kind of mentoring communications and some of the public service hours. And so let's come back at 10:40 and then finish the second half of the session. Thank you.